Hey, hello students! We talk about these differentiation markers, but what are they exactly? Let's see this together. Now, we will take two examples. The first one is muscle cell differentiation. I take this one because it's pretty easy to understand and it is a clear illustration of differentiation markers, but the same principle applies to any type of differentiated cell. So here we will start with the mesenchymal cell. It is in fact the progenitor cell for muscle cells, but not only for muscle cells. Mesenchymal cells are progenitors for fat cells or adipocytes, for cartilage cells or chondrocytes, and for muscle cells or myocytes. So the mesenchymal cell will divide and differentiate. The differentiation occurs under the control of specific signals that come from the exterior, so it can be dissolved molecules like growth factors, or it can also be signals from the extracellular matrix, so molecules in the environment of the cell that will contact the cell and induce a signal of differentiation. After several rounds of cell division and differentiation, the mesenchymal cell will produce a lot of myocyte progenitors, so cells that will really become myocytes and muscle fibers, but they will also produce a small proportion of cells that will keep a capacity to divide, so these ones will be muscle stem cells. So they will stay in the pool of cells even in adults so that the muscles can renew and regenerate. What is interesting to distinguish is, first of all, the shape of the myocyte progenitors and the shape of the muscle stem cells. In fact, we can differentiate them because they have a different appearance. Not only the shape, actually, in fact, their intracellular composition is also slightly different. In addition to that, we can also distinguish protein expression that is different in the myocyte progenitors versus in the muscle stem cells. So among the proteins that are differentially expressed, we have the MyoD protein. So this protein identifies specifically the myocyte progenitors. If you have a cell with MyoD in it, it is a cell that is oriented towards becoming a myofiber. In contrast, if you have the expression of the PAX7 protein, it is a protein that identifies the muscle stem cell. So it's pretty easy to tell apart the myocyte progenitors from the muscle stem cells by looking at their proteins. No MyoD and no PAX7 means that it's a non-differentiated mesenchymal stem cell, so not yet oriented in the muscle lineage. If you have a MyoD positive cell, it is a myocyte progenitor, so it will form muscle fibers. If it is a PAX7 positive cell, it is a cell that will stay in the pool of muscle cells, but in the form of a satellite cell, which is actually the stem cell of muscles. So these are differentiation markers because they allow us to identify cell types based on their presence or absence. Other differentiation markers can include the shape of the cell or the presence of intracellular structures like contractile fibers. So now that we understand the concept of differentiation markers, let's have a look on differential gene expression. If we take this time an example with adipocytes or fat cells, again, we start with the same mesenchymal stem cell like the one we had for myocytes. We see that before it differentiates between fat and muscles, there is a first step of differentiation. Some cells will be MIF5 positive and others will be MIF5 negative. The MIF5 positive cells can either become myocytes or brown adipocytes, and the MIF5 negative cells can become only white adipocytes or white fat cells. Now it is important to understand the difference between a brown and a white adipocyte. A brown adipocyte is a fat cell that has a thermogenic function. It is present essentially in hibernating animals or in newborns and up to maybe 2 years old. But after that, in human adults, the majority of adipocytes we have are rather white adipocytes, which don't have a thermogenic function, but they have a fat storing function. So they have an enormous fat droplet, and the fat droplet is so big that it squeezes the nucleus on the side of the cell. So now, understanding this difference, we can understand a little bit better what the signals are and what they do. So if you look here, once you have your MIF5 positive cell, if it is exposed to male sex hormones, thyroid hormone, or cold, then it will differentiate into a cell that has little fat stores in small droplets and that mostly has a lot of mitochondria to be able to produce heat and energy from fat. In contrast, a cell that is MIF5 negative and that is exposed to stress hormones or to insulin, which has the goal to remove sugar from the blood, then this pre-adipocyte will further differentiate into a white adipocyte. So it is possible to have a MIF5 negative cell that never really becomes a fat cell just because it's not exposed to these signals, but as soon as it becomes exposed to them, it will differentiate into the pre-adipocyte and then the white adipocyte, and it will build its droplet pretty quickly and store fats in this droplet. So the signals that can be varied will induce differential gene expression so that from the same MIF5 positive cell you don't have a myocyte but you have a brown adipocyte, and these genes encode proteins supporting the thermogenic function, while in this case it is rather proteins that support the adipogenic function. So for instance, enzymes that convert sugar into fats. So I hope that now the concept of differentiation markers and differential gene expression is a bit clearer. And remember that a differentiation marker is anything that allows you to identify the cell, and it's anything that is unique to the cell you want to identify. It can be anything, including the shape, the intracellular composition in filaments, the quantity of a certain type of organelles, or the expression of a specific protein or a subset of enzymes. All that can constitute differentiation markers. And they are the visible effects of differential gene expression. This is it for today. Bye!